So my talk will be centered mostly on the genetic variation in NAT2, as well as some of the GST genes across sub-Saharan African populations, with a specific focus on how it can influence tuberculosis treatment. Drug response varies from person to person. If all of us in this room were given the same dose of the same medication, some of us will respond favorably. However, some individuals may experience some adverse reactions or experience no therapeutic benefit. The variability in drug response can be attributable to several factors, such as age, sex, the microbiome, as well as genetics. My research is in the context of pharmacogenomics, which deals with understanding the influence that genetic variation has on drug response. The future goal of PGX research is to essentially move away from a one-size-fits-all approach to drug administration and to rather use an individual's pharmacogenetic profile to inform safer treatment choices, which may include altering the drug dosage or choosing a more suitable drug to avoid side effects while promoting efficacy. Currently, there are relatively fewer PGX studies in Africa compared to the global north. However, the field is slowly gaining traction on the continent. Here, I'm highlighting two recent African PGX projects, and my study is under the Project Africa Gradient Initiative, which aims to uncover the impact of genetic variation on response to TB and malaria drugs. My project focused more so on TB treatment with isoniazid because of the serious side effect of drug-induced liver injury. Isoniazid is one of the commonly prescribed first-line TB treatments. This slide highlights proteins that are important in both phase 1 and phase 2 metabolization of the drug. Several PGX studies have shown that variation in the encoding genes can influence an individual's response to the medication. The genetic variation in these pharmacogenes are considered as haplotypes, which are represented by star allele nomenclature. A star allele differing from the reference haplotype is typically defined by various combinations of non-synonymous variants, as shown by these two star alleles for NAT2. But in some cases, star alleles can also be defined by structural variants, such as the deletion of the GSTM1 gene, represented as GSTM1 star 0. A major challenge in generating comprehensive evidence to support broader anti-TB pharmacogenetics testing is the scarcity in the information regarding the distribution of star alleles as well as their impact on drug response, especially across a continent such as Africa with a high genetic diversity and where TB and similar health burdens are very prevalent. Fortunately, there's increasing availability of genomic data from African populations, which provides an opportunity to address the gaps in PGX diversity in Africa, especially among underrepresented population groups. This study, therefore, aimed to leverage the recently generated African whole genome sequencing datasets to characterize the distribution of star alleles in 6 pharmacogenes across diverse African populations. Understanding the genetic landscape in these genes could inform the prioritization of variants for further functional studies, which is important in informing possible optimization of drug dosage regimens for drugs such as isoniazid or even other anti-TB drugs, with the hope of mitigating adverse drug reactions such as drug-induced liver injury. This was a comparative study entailing secondary analysis of high-coverage whole genome sequencing data from various sub-Saharan African populations. A total of 1,079 participants were included, mainly from the H3 Africa projects. The dataset also included high-depth Africa genomes generated from the Simon's Genome Diversity Project, as well as the 1,000 Genomes Project. This comparative analysis was extended to compare the distribution of star alleles that were called in the SSA populations with 6 other global populations stemming from the 1,000 Genomes Project. Stellar PGX, a bioinformatics pipeline that was developed by our group at the University of the Witwatersrand, was used to call star alleles in these pharmacogenes that were analyzed in the study from the whole genome sequencing data. In the instance of novel star alleles, the defining variant or variants were annotated and a functional prediction was made using in silico prediction tools. The phenotype prediction was based on combining the associated activity of the haplotypes that form an individual's diplotype for that particular gene. In regards to the statistical analysis, we use the Hardy-Weinberg equilibrium test to see whether the frequencies that we observed differed from the expected frequencies, while using the Fisher's exact test to see if there was any statistical significance regarding the differences in the frequencies that we observed between the different population groups. This is a stack plot showing the NAT2 star allele distribution in populations from Western, Central, Southern, and Eastern Africa. There are considerable differences in the star allele frequencies when comparing some of the Western African populations to that of the Southern African populations. Even as you look at the Southern African populations alone, there are differences in the frequency of NAT2 star alleles, such as star 1, star 4, as well as star 5. The difference in the star allele distribution also translates to the differences in the phenotypic distribution for NAT2 across the different SSA populations. The overarching prevalent phenotype was the intermediate acetylator, shown in green. Populations of African ancestry had a similar NAT2 phenotypic profile. However, this differed in comparison to the other remaining population groups that were analyzed. There was a difference in the distribution of the GSTM1 as well as GSTT1 gene deletion. In the SSA as well as African American Afro-Caribbean populations, the GSTT1 star 0 was relatively higher compared to the GSTM1 star 0. An opposite observation was seen for the other global populations. Again, the phenotypic profile for the other genes, for both of these genes, differed across the different population groups. Notably, the GSTM1 no-function phenotype, shown in the lighter blue, seemed to be more prevalent compared to the GSTT1 no-function phenotype, shown in the same color. There were 47 potential novel haplotypes that were identified across these anti-TB pharmacogenes, and I've just listed some of them. 26% of these haplotypes had defining variants that were predicted to be deleterious. One of the NAT2 potentially novel haplotypes were identified in the 1000 Genomes Project dataset, which was further resolved using TRIO data and has since been cataloged on the PharmVar database. In this talk, I've described the variation in these genes within the context of TB treatment, but it's important to note that these genes are also responsible in the metabolization of other drugs for the diseases that are shown on the slide. Therefore, the impact that genetic variation has also needs to be considered for these drugs. In terms of the limitations, our population demographic largely consisted of the ethno-linguistic groups from the Niger-Congo language family. In addition, the smaller subgroups of the SSA populations were relatively small. Some of the genes lacked a curated and comprehensive star allele definition, which made it difficult for us to compare the haplotypes that were called within our study to those of the previously to those that have been previously published. We also used short read sequencing data, which made it a bit difficult to phase some of the variants that were defining these novel haplotypes. However, where possible, we did use TRIO data or use statistical phasing methods. Functional studies to understand the clinical impact that these novel haplotypes have in terms of drug response were outside the scope of the study. Lastly, the phenotype predictions that were made in the study were based solely on the genetic variation, and therefore the actual observed phenotypes that an individual may present with may be influenced by other factors such as age, diet, or even environmental factors. Pharmacogenomics analysis is important in the African context because of the vast genetic diversity and varying drug response profiles. The predicted allele and phenotype distributions in our research show that variation in NAT as well as GST genes might contribute to known adverse drug reactions such as drug-induced liver injury. Future work would involve obtaining a more complete spectrum of PGX variation in Africa through the development of bioinformatic tools or fostering collaborations with other researchers who have access to African genomes. In addition, functional as well as clinical studies can aid in understanding the impact that variation has on drug response. I'd just like to acknowledge my two supervisors, the principal investigators of the respective datasets, the Project Africa grading collaborators, as well as the funders. Thank you.
What is the ratings beyond that? For example, the guy has the treatment beyond what and honestly, we don't understand that from the treatment. But sometimes it's difficult. So what can we do to comment the drug mechanism the way
Okay, so to your first question, so I know, for example, with NAT2, it's split into 3 different phenotypes. So you have your slow, intermediate, and rapid acetylators. So it's been shown that with slow acetylator, or people who are slow acetylators, they tend to develop this drug-induced liver injury more so than an individual who might acetylate the drug on an intermediate level or even on a rapid level. So that's what I've seen in terms of like the literature. And then for your second question was about like translating what we have here. So I think, how would I explain this? Okay, so I think in terms of, there's certain phenotypes that probably would guide you to know whether you need to optimize the drug dosage, so whether to increase it or decrease it. So for example, with the GST genes, those are more responsible for the detoxification of the drug. So obviously, if you have one copy of that gene not being present, then you would assume that you need to probably give that individual a lower dose. But now, if you don't have any of those copies, and if those are the only like enzymes involved in the detoxification process, then you'd have to essentially look at an alternative drug or find another way to kind of manage the side effects that an individual may experience. Okay. So to answer your question, no. So this research was done on healthy individuals. So we were just looking at what is the variation that is present within the genes that we know are involved in the metabolization of that drug. But I do understand that looking at a drug-resistant strain and seeing if we can also kind of include that in a way to try and understand how someone might not necessarily metabolize, but respond to the drug, whether they will, whether the drug will be effective or not, is obviously an important factor, but that's not something that we looked at here. Yeah. Okay. So that is something that is worth looking at, trying to develop like a genetic test to be able to give an individual kind of like their pharmacogenetic profile. That's not something that I have done, but I know that that would be useful. And I think it's also been mentioned in the prior talks how genetics is kind of like a static thing. So once you've tested someone, then you know that, okay, this is their genetic, and you can kind of aid in treatment dosage, like, you know, so yeah. No problem. Mhm. Yes.
knowing that people is as possible, have you considered only looking these that call
so for specifically for isoniazid, there is only one CYP450 gene, which is the CYP2E1, that's involved in the metabolization. Unfortunately, it's also characterized as one of those genes that don't really have a well-defined kind of like nomenclature or for the star allele. So it's kind of difficult to be able to like, let's say, for example, you test for those variations, you can't really say, okay, because you have this variant, this is your phenotype, because those studies are not very well standardized, if you can put it in that sense. But it is something that, yeah, you need to kind of still test for so that you know, okay, this is maybe what could be the issue in regards to like having an adverse reaction. Okay, so there wasn't really a defined method as to why we chose one data set over another. I think the main focus was looking at countries that fall under the like Sub-Saharan African region. So there's not really a method to that. And yeah, the numbers were just the numbers that were there in those respective data sets. So it's kind of like we had to work with what we had. But I think even if it is if one population might have been smaller than another one, I think it's still important to kind of do a discovery analysis just to see what's out there.